Stem cells are undifferentiated cells with the remarkable ability to self-renew and develop into specialized cell types. This field holds immense promise for medical applications, as stem cells could potentially be used to regenerate damaged tissues and treat a wide range of diseases. The image on this slide shows a close-up view of a stem cell dividing in a petri dish, with a vibrant color palette and soft lighting that highlights the dynamic nature of these cells. I'll provide an overview of the key characteristics of stem cells and discuss some of the exciting developments and challenges in this rapidly advancing field. Stem cells are a unique type of cell with remarkable properties. They have the ability to self-renew, dividing repeatedly while maintaining an undifferentiated state. Stem cells are also multipotent, meaning they can give rise to various specialized cell types within a tissue or organ. This differentiation potential allows stem cells to transform into specific cell types like muscle, nerve, or blood cells, with unique functions. Embryonic stem cells come from the inner cell mass of a very early-stage embryo, called a blastocyst. They are pluripotent, meaning they can develop into any cell type in the body. Adult stem cells are found in various tissues like bone marrow, skin, and brain. Adult stem cells are multipotent, meaning they can only differentiate into a limited range of cell types specific to their tissue of origin. The key difference is the breadth of cell types each type of stem cell can become. Embryonic stem cells are a special type of cell found in the early stage of a developing embryo. These cells have the remarkable ability to transform into any type of cell in the human body, from brain cells to muscle cells, and everything in between. This makes embryonic stem cells incredibly valuable for medical research and potential treatments, as they could be used to regenerate damaged tissues and organs. The image shows a microscopic view of these stem cells, highlighting their unique structure and the process of cell division and differentiation. The vibrant colors and glowing effects help visualize the dynamic and complex nature of these remarkable cells. Adult stem cells are found throughout the body in various tissues. They have a limited ability to self-renew and differentiate into new cell types. They are multipotent, meaning they can become a limited number of cell types within their tissue of origin. For example, blood stem cells in bone marrow can form different blood cells, and brain stem cells can generate neurons and glial cells. These adult stem cells play an important role in tissue repair and regeneration. Induced pluripotent stem cells, or iPSCs, are adult cells that have been reprogrammed to behave like embryonic stem cells. This reprogramming process involves introducing specific genes or proteins into adult cells, causing them to revert to a pluripotent state. In this pluripotent state, iPSCs can then differentiate into a variety of specialized cell types. The ability to generate iPSCs from adult cells is a major breakthrough, as it avoids the ethical concerns associated with using embryonic stem cells. iPSCs have tremendous potential for regenerative medicine, disease modeling, and drug discovery applications. The detailed microscopic image on this slide shows the intricate structure of an iPSC, with a nucleus, cytoplasm, and other organelles clearly visible. This level of detail highlights the scientific wonder and complexity of these remarkable cells. Stem cells have the unique ability to develop into various specialized cell types, which is known as their potency. The level of potency varies, ranging from totipotent, can become any cell type, to pluripotent, can become many cell types, to multipotent, can become related cell types, to unipotent, can only become one cell type. A stem cell's potency determines the range of cell types it can give rise to, which is important for its role in development and potential therapeutic applications. Understanding stem cell potency is crucial for harnessing their capabilities in fields like regenerative medicine and tissue engineering. Totipotent stem cells are the most versatile type of stem cell, with the ability to differentiate into all cell types, including the placenta and other extraembryonic tissues. They're found in the early stages of embryonic development, specifically the zygote and the first few cell divisions. These cells have the exceptional developmental capacity to give rise to a complete organism. Totipotent stem cells have unlimited potential, demonstrating their remarkable versatility and importance in the early stages of life. Pluripotent stem cells are remarkable for their ability to differentiate into any cell type in the body, except for the placenta. This unlimited differentiation capacity makes them incredibly valuable in regenerative medicine. Pluripotent stem cells are found in the inner cell mass of a blastocyst, the early stage of an embryo. They are responsible for the development of all the tissues and organs of the body. Pluripotent stem cells hold immense promise for treating various diseases and injuries. They can be used to generate new cells, tissues, and even organs, offering hope for patients with conditions like spinal cord injuries or Parkinson's disease. Multipotent stem cells are a special type of cell that can differentiate into multiple cell types within a specific lineage. These cells play a crucial role in the regeneration and maintenance of tissues in the body. Mesenchymal stem cells, MSCs, are a prime example of multipotent stem cells. They are found in the bone marrow and other tissues and can differentiate into bone, cartilage, fat, and other connective tissues. Neural stem cells, NSCs, are another important type of multipotent stem cell. They are found in the brain and spinal cord and can generate neurons, astrocytes, and oligodendrocytes, contributing to the development and repair of the nervous system. Multipotent stem cells have immense potential for regenerative medicine and tissue engineering, as they can be used to replace or repair damaged or diseased tissues. Unipotent stem cells are the most specialized type of stem cell. They can only differentiate into one specific type of cell. This allows them to maintain a steady supply of differentiated cells in the body. Examples include muscle stem cells, blood stem cells, and nerve stem cells. These stem cells have a very limited potential compared to other types like pluripotent stem cells. Their role is to replenish and repair the specific tissues they are associated with. The stem cell niche is a specialized microenvironment that supports stem cell survival, self-renewal, and differentiation. It provides a complex interplay of physical and chemical cues that regulate stem cell fate. The key components of the stem cell niche are: extracellular matrix provides structural support and signaling molecules. Neighboring cells communicate with stem cells through direct contact. Growth factors regulate stem cell proliferation and differentiation. Oxygen concentration affects stem cell metabolism and function. These factors work together to create a precisely controlled environment that enables stem cells to maintain their unique properties and respond to developmental and environmental signals. Stem cells have the remarkable ability to divide and produce daughter cells that maintain their undifferentiated state. This self-renewal process ensures a continuous supply of stem cells, which is crucial for ongoing tissue regeneration and repair throughout life. A complex interplay of intrinsic factors like genes and signaling pathways, as well as extrinsic factors like the stem cell niche, regulate this self-renewal process. Self-renewal is essential for maintaining tissue homeostasis and enabling regeneration after injury or disease. This slide introduces the key stages of stem cell differentiation, the process by which stem cells transform into specialized cell types. The first stage is commitment, where stem cells commit to a particular lineage like blood, muscle, or nerve cells. This involves turning on and off certain genes. Next is specification, where the committed cells acquire more specialized features and become further restricted in their developmental potential. The final stage is maturation, where the cells fully develop their functional characteristics and become ready to perform their specific roles in the body. The visual on the slide helps illustrate these stages, showing the transition from a stem cell to a mature red blood cell. I'll use this timeline to walk through the key steps of stem cell differentiation and how cells progress from a pluripotent state to a fully specialized state. WNT signaling is critical for stem cell self-renewal and proliferation, regulating the expression of genes essential for maintaining the stem cell state. Hedgehog signaling plays a crucial role in stem cell fate determination, influencing the differentiation pathways that stem cells can take. Notch signaling is essential for cell-cell communication, regulating the balance between stem cell self-renewal and differentiation to ensure proper tissue development. TGF-beta signaling is involved in stem cell maintenance and differentiation, regulating the expression of genes controlling cell cycle progression and differentiation. Transcription factors are key regulatory proteins that control gene expression in stem cells. They act as master regulators, binding to DNA and turning genes on or off. This allows them to determine whether a stem cell remains undifferentiated or differentiates into a specific cell type. Combinations of transcription factors create complex networks that precisely control stem cell fate and development. Understanding how transcription factors function is crucial for understanding and directing stem cell behavior. DNA methylation is a key epigenetic mechanism that can regulate gene expression in stem cells. Methylation patterns can be dynamically altered, contributing to the control of stem cell self-renewal and differentiation. Histone modifications, such as acetylation and methylation, play a crucial role in regulating chromatin structure and accessibility to transcription factors, thus influencing gene expression in stem cells. Non-coding RNAs, including microRNAs and long non-coding RNAs, can influence gene expression by targeting specific mRNA transcripts, affecting the fate of stem cells. Stem cell plasticity is the ability of a stem cell to differentiate into a different cell type than its original lineage. This challenges the traditional view of cell fate determination, which held that stem cells were more limited in their differentiation potential. Some studies have found that stem cells from one tissue, like bone marrow, can differentiate into cells of another tissue, like muscle or nerve cells. This could have important implications for regenerative medicine and treating diseases, as it suggests stem cells may be more versatile than previously thought. However, this is a controversial area of research, and the full extent of stem cell plasticity is still being investigated. Stem cell therapy is a revolutionary medical approach that harnesses the incredible regenerative potential of stem cells. By injecting stem cells directly into damaged tissues or using them to grow new tissues for transplantation, we can treat a wide range of diseases and injuries. The applications of stem cell therapy are truly exciting, from helping patients with spinal cord injuries regain function, to regenerating damaged heart tissue, to potentially even treating diabetes. While the science is still evolving, the promising outcomes we've seen so far make stem cell therapy a field of great hope and promise for the future of medicine. Regenerative medicine is an exciting and rapidly evolving field that holds great promise for improving human health. It harnesses the incredible potential of stem cells to repair and regenerate damaged tissues and organs. This approach aims to treat previously untreatable diseases and conditions by replacing damaged cells or tissues. While still an emerging field, ongoing research and clinical trials are refining the techniques and expanding the applications of regenerative medicine. The hope is that this field will restore function and reduce disability, dramatically improving quality of life for many people. Tissue engineering is a field that combines cells, scaffolds, and biomaterials to create artificial tissues and organs. Cell culture is the foundation, where we carefully grow and maintain the necessary cells in a controlled environment. Scaffolds provide structural support for the growing cells, mimicking the natural extracellular matrix and giving cells a framework to attach and proliferate. Biomaterials are used to create these scaffolds and other components of engineered tissues. They must be biocompatible and support cell growth. Tissue engineering has numerous applications, including the development of artificial skin, cartilage, and even organs to replace or repair damaged tissues. Stem cell transplantation is a promising medical approach for treating various diseases and injuries. Transplanted stem cells can differentiate into specialized cells, replacing damaged tissues and restoring organ function. Extensive research and clinical trials are ongoing to evaluate the safety and efficacy of stem cell transplantation. Ethical considerations around stem cell transplantation include informed consent, donor selection, and potential risks and benefits. It's an exciting and rapidly evolving field with the potential to transform how we treat many serious conditions. Stem cell banking is the process of collecting, processing, and storing stem cells for potential future use. This allows individuals to have access to their own stem cells, which could be used for future therapeutic purposes. There are two main types of stem cell banks, public and private. Public banks collect and store stem cells from umbilical cord blood, which can be used by other individuals. Private banks store stem cells specifically for the individual who donated them. Stem cell banking provides a way to preserve these valuable cells for the future. Stem cell research holds great promise for treating a wide range of diseases, but it also raises important ethical considerations that we must carefully navigate. One key concern is the use of embryonic stem cells, which involves the destruction of human embryos. This raises profound questions about the moral status of embryos and the ethics of sacrificing them for research. Another critical issue is ensuring informed consent, especially when working with vulnerable populations who may not fully understand the implications of donating their stem cells. The potential for commercialization and profit-making from stem cell therapies is also a concern, as we must ensure equitable access to these treatments and prevent them from becoming the exclusive domain of the wealthy. Finally, the ability to genetically modify stem cells raises worries about the unintended consequences that could be passed down to future generations. We must proceed with great caution and foresight in this area. Overall, stem cell research offers immense potential, but we have a responsibility to carefully consider the ethical ramifications and find ways to harness this technology in a manner that is ethical, equitable, and protective of human dignity. International guidelines from organizations like the ISSCR provide ethical and scientific frameworks to ensure responsible stem cell research practices worldwide. Each country has its own regulatory framework for stem cell research, covering ethical considerations, safety protocols, and clinical trial approvals. Institutional review boards, IRBs, evaluate the ethical and scientific merit of stem cell research proposals involving human subjects to safeguard participant welfare. Regulations govern the collection, storage, and distribution of stem cells, ensuring quality control and responsible use of these valuable resources. Stem cell therapy holds great promise, but there are several key challenges we need to address. One major issue is immune rejection. Stem cells from donors can trigger an immune response in the recipient, making the therapy ineffective. Another concern is the risk of tumor formation. Undifferentiated stem cells can potentially form tumors, which is a serious safety risk. The use of embryonic stem cells also raises ethical considerations around the destruction of human embryos. Finally, stem cell therapies are expensive and not widely accessible, limiting their use to many patients who could benefit. Overcoming these challenges will be critical to realizing the full potential of stem cell therapies. The future of stem cell biology is full of exciting possibilities, from advanced screening techniques to personalized therapies. Developing more accurate and efficient methods for screening potential stem cell therapies will be crucial to ensure safety and efficacy. Tailoring stem cell therapies to the specific needs of individual patients will allow for more personalized and effective treatments. Ensuring the long-term safety and effectiveness of stem cell therapies is a top priority and will require ongoing research and collaboration. Fostering collaboration among researchers, clinicians, and ethicists will be essential to advancing the field of stem cell biology and realizing its full potential. Stem cell research is a rapidly advancing field, with new discoveries constantly expanding our understanding and potential applications. While stem cell therapies hold great promise, there are ongoing ethical debates surrounding certain stem cell sources, such as the use of embryonic stem cells. It's important to acknowledge these ethical considerations and engage in thoughtful discussions to address them as the field continues to evolve. The key takeaway is that stem cell biology is a dynamic and exciting area of study, but we must navigate it carefully, weighing the potential benefits against the ethical concerns.